From the analytical point of view, I want to make a short, quick clarification of some background or some terminology, especially the so-called low-dimensional embedding or dimensionality reduction, which is now the indispensable component in analysis of single-cell data. Just to make sure we're at the same page. So when we refer to low-dimensional embedding, we mainly mean that we want to find a low-dimensional representation of this high-dimensional data set of, say, gene expression from single-cell RNA sequencing reads. And here we apply a nonlinear dimension reduction method, t-SNE, to a typical experimental data. And the low-dimensional embedding essentially characterize the cell-cell similarity, or just a low-dimensional representation, so that we hope that the geometric pattern or some underlying feature of the data is well preserved in such a low-dimensional presentation. And here we can see there are cluster patterns, which turns out to capture the cell type information. Of course, we do not expect it's the only information we can get from this high-dimensional data set. But the low-dimensional embedding normally plays an important role in carrying on the low-dimensional, the intrinsically low-dimensional information about this high-dimensional data, and it plays a role in many downstream analysis as an instrumental tool. So very important challenge in current single-cell analysis is the large availability of data sets coming from all different sources about different samples or different experimental platforms about tissues or organs, species, and so on and so forth. And basically, for every day, there are tons of data generated from labs all over the world. And because of the effort of the open source repository and databases, most of these data sets are becoming more and more available. So that for a typical biological problem of interest, we don't normally we don't just have one data set to work on or to look at. We have multiple data sets, and the idea is that by interpretive analysis of multiple data sets, we will hopefully get a more comprehensive or unbiased profiling of the biological process of interest. And the challenge here would be how to enable the integration across multiple samples, multiple data sets of very heterogeneous nature. And here I'm going to show you 3 basic examples where integration are needed. So first example is cell atlas projects. Our human body is a mixture of different kinds of cell types. Even for different organs, it may contain different types of cells as a composing part. And one interesting problem is to obtain a high resolution map of single cell types, so that we can have a global view of how many cell types consist of there and what are their interactions or relationships. And of course, it's very difficult to get a single sample from our body that contains all different cell types. So we need to combine many samples corresponding coming from different organs, let's say. And one interesting problem would be to identify the shared or distinct cell types across different tissues or different organs. And in order to do so, we need to integrate data set, basically samples corresponding to different organs. Okay. And another need of integrating multiple samples is that for many cells, they are so rare, less representative in a single sample. So to enable our detection or the power of detection, we really need to have large number of cells, which only comes from integrating multiple samples. So here, the integration in this context would mean we want to aggregate multiple samples together. Here is just an example of a recent study that enabled the integration of cells coming from multiple organs, so that eventually they got this map of, in total, 51 different cell types. Here, each dot is one sub-cell type defined, and the colors are just the 12 major cell types they classified. And in this embedding, the distance of different cell types would reflect their similarities, basically. And second, in the second example, when we sometimes we want to have a understanding of disease-associated cell states. So basically, we not only do we have different cell types, but for each cell type, it may contain or may have different cell states under different conditions, like disease and normals. And sometimes, as you can imagine, disease can cause sort of creation of new cell types that have a very abnormal behavior. And one problem of interest would be to identify how the cell states got altered by some specific diseases. And in order to do so, it is not just sometimes it's not enough to just look at the a sample that comes from a patient. It is useful to have a comparative analysis by using both normal and disease tissues, so that by doing some correspondence analysis, we would be able to identify some disease-specific cell states. And in this case, the integration would really means that we want to have a cross-condition or case control comparison of different samples. Well, you may wonder that why this is even a challenge. One can just use, say, standard tools from statistics to sample tests to see the differences. But shortly we will come back to this, just to explain that there are some intrinsic technical barrier that prevent us from doing so. And in this case, again, the challenge is to enable comparison across different samples. And this is just an example of recent study that exactly follow this strategy by comparing normal and disease tissues to identify some disease-specific cell states. And the third example is basically the single-cell multi-omic research for, say, let's say, cancer biology. And in this case, for each individual cell, we not only have a measurement of one modality like gene expression or chromatin accessibility, we can have more advanced technology so that multiple molecular features would be captured for individual cell. For example, here we basically from this manuscript is just from one of our collaborators at Cold Spring Harbor. They recently developed a sequencing technology, basically for each individual cell, instead of just returning a matrix of gene expression for cells, they can obtain two matrices. One is, again, the gene expression profile. The other is actually the DNA copy number profile for the individual cells. So for cancer studies, as we know that DNA and RNA are highly involved in the biological processes, and now we have the measurement of DNA and RNA of the same cells. For tumor samples, we will be able to have a better understanding of the interaction of DNA and RNA. And here, the integrative analysis really meaning that for the same cell, we want to aggregate different kinds of features. Right? One is RNA, the other is DNA. They have some correspondence, but not identical, of course. So here, integration would be meaning that enable feature aggregation across different modalities.
What's the name of this paper? Is this a successor of GNTC?
Yes, this one they call BAG-seq. It's from the lab of Michael Wigley.
BAG-seq?
Yeah, BAG.
B-A-C?
B-A-G.
Okay.
Yeah.
Interesting. How many cells are they able to?
Yeah, for typical sample, they can get thousands. Like, yeah, that's advantage is high throughput. Yeah. Yeah, so briefly summarizing what we just talked about, actually we can classify the current integration tests into three major categories. And in the first category, which include our examples 1 and 2, the integration would mean that we have basically identical features, like a list of common genes or a list of, say, peaks from chromatin accessibility or whatever. Basically, it's identical features, but different samples. We want to aggregate samples together. And in this case, we can call it a data alignment. We want to align the samples with under the same features. The second type of integration here we have, which basically covers the third application, the multi-omic analysis. with paired measurement. In that case, it's about the same samples, but different kind of features. And in this case, it's more or less known in machine learning literature as multi-view learning, that we have different views of the same object, but how to integrate the information from different views. In the third type of integration task, which is more challenging, which is a kind of mixture of the previous two, sometimes also known as multi-omic study with unpaired data. You have a measurement of gene expression for a bunch of cells, but they copy number variation for another bunch of cells. There is no natural correspondence between the cells, so you have to either computationally make the correspondence or even using some other experimental strategies. But in this case, we basically neither have paired features in our cells. So we could generally call it multi-model integration. It's a more challenging task. And in this talk, we're going to talk about focus on the first type of integration. And if time allows, we can mention how it is possible to extend to other type of integration. Okay, now come back to the original question, why it is a challenge to integrate multiple samples. Why not just merge data together? So the challenge comes from this technical barrier, essentially comes from the single cell experimental design. Basically, from the output of a single cell experiment for multiple data sets, each coming from associated with either one study or one sample, even after mathematically normalizing each data set, it turns out that the entries in each data matrices are still not comparable across samples. So basically, there are so-called batch effect that does not go away even if you normalize each data set. And here we look at this example. Here we have two data sets. Each is RNA sequencing read for some blood cells. It's same experimental strategy, but just from two samples. Okay, and we can see that there is a significant batch effect between the two data sets. Here, red corresponding to the data set 1, black data set 2. Again, we can see the between data set differences are of almost the same magnitude of the within data variations. So if we just simply merge to do the two data sets together, we won't be able to have enough power to identify any within data variations. But even for this particular example, if we just examine this visual appearance, we can see that by focusing on one color, there are some kind of low dimensional or just visually some structure in the data set. And the low dimensional structure seems to be shared between the two data sets. It seems like there are some underlying transformation that is going on that makes the difference or discrepancy between the two data sets. And just by this visual conception, we can consider the batch effect to be whatever caused the differences between the two structures that make the differences. And we can imagine that some analytical strategy might be possible to just remove whatever existed, just the gap between the two data sets. And it turns out that by conceiving this problem as a manifold alignment, that for each data set, there are some low dimensional structure. And we want to just identify that low dimensional structure and remove the differences between, or just align the low dimensional structure within two data sets. It turns out to be a not very difficult machine learning task. And many algorithms has been developed just to remove the so-called batch effect so that you have perfect alignment of the two data sets. And here we apply one of the popular machine learning algorithm. It turns out that just after this procedure of the differences between the so-called batch effect between the two data sets completely removed, then we see a perfect alignment of the two data sets in terms of that embedding. And yeah, so here are just the three most popular algorithms that are currently available. There are more than that, as we will see shortly, but these are arguably the most popular ones. And it's just came out a few years ago and immediately raised dramatic attention over within the community and made profound impact to the subsequent scientific discoveries. From that, we can actually make two observations. First of all, this data integration step actually plays a pivotal role in the current analytical pipeline. So as I said, we don't just start with one sample for typical experiment or typical study. So there more or less always will be a step in the analytical workflow so that we need to integrate multiple samples. And secondly, because of this step happens relatively early in the whole analytical pipeline, so it have profound impact for all the subsequent analysis. Basically, the data integration procedure, right or wrong, it may have impact to the subsequent analysis. Some of them can be very serious. And because of this importance, people all realize that it's just something essential in current single cell analysis. So now over 60, let's say a conservative number, over 60 integration methods that are now available in our Python or some other things. And there are even studies now just dedicated to compare the pros and cons of integration algorithms in terms of different criteria or different downstream tasks. And we can consider the leaderboard of different integration algorithms to evaluate them. Despite the diversity of different integration algorithms, most of them seemingly relies on a common assumption or heuristic to perform integration. So the key assumption underlying most of the algorithm is as follows. So that there exists some common low-dimensional structure. It could be cell type information or cell states information or some other things. Essentially, it has to be something low-dimensional that can be captured by some dimension reduction algorithms or manifold learning algorithms. And the algorithm internally first capture that common low-dimensional structure and then try to perform an alignment of the corresponding parts. So that in the end, they want to find a comprehensive matching. Basically, everywhere got one-to-one correspondence. And whatever that can be accounted for by this transformation got completely removed. However, despite its power in many existing analyses, now from the community, people start to reflect that whether these assumptions really hold for every single analysis that we are currently encounter. First of all, does this assumption always hold? Meaning that for any data set that we want to integrate them together, is that really true that they all contain the same low-dimensional structure of interest? The second question would be, is this comprehensive matching always what we need? Right? For two data sets at hand, we want to integrate them together. Do we always want that whatever differences between the two data sets that has to be overcome after this integration? Right, please.
So is that assumption also coupled with the fact that the experiments need to be the same? Like that's right, yes. Because like if the design is
it really depends on the biological question of interest. Like in the second example, if it's just the identification of differential structures between the samples, although there are some batch effect that you need to overcome, but it's not the case that you just want to overcome all the differences, right? You want to have some cell states, differential cell states altered by disease to be identified in the end. But some you will see it. Sometimes the integration algorithm is just so overconfident so that it will wipe out everything. So Yeah, so again, more recently, people just start to reflect on this whole practice based on integration algorithms, and some authors find it's a serious issue about the whole practice of many downstream analysis building up on the integration, and some of them they are worried about ignorance of some just technical issues that may happen during the integration, which may have impact in the downstream analysis, and even in making scientific conclusions. So now let's just review what they discovered about the limitations of current integration algorithms. Okay, so the first limitation, as I just mentioned, that most existing methods seems to be over-optimistic about this task of integration. Basically, there is a risk of overfitting, or just in order to make the alignment, it substantially distort individual data set in order, as I said, in order to make the comprehensive alignment matching between two data sets. So now we just did a very simple experiment. We got two data sets where we know that there is completely no correspondence between them. It's just two types of tissues with no overlapping cell types, right? And then we applied some aforementioned integration algorithm to see what happens. And it turns out that the algorithm still, if you ignore the cell type annotations, just look at the embedding and alignment, you can see they still did reasonably well in making them together. Okay, and after you're evaluating the cell types, you can see there's a significant misalignment or distortion, so that different cell types got collapsed together across data sets or even within data sets. And this just motivate us to think that for many existing methods, although they did pretty well in many benchmark studies, they don't provide information about when we should or shouldn't integrate two data sets together. Okay. Related to that, a second limitation is that many existing methods seems to be black box algorithms, where we just take many data sets to be integrated together as the input, and the output is just the integrated data set, where we don't know what have been done, what process have been made into this integrated data set, except for this is the outcome. And especially most of these algorithms, they call themselves batch effect or batch effect correction method, but it is unclear from case to case what does it mean by batch effect, right? Sometimes the batch effect can vary from one application to the other, depending on what you need. Sometimes batch effect may not just be the technical variations created by the sequencing procedure. It could be some biological variations. It's just something that you don't want at the current stage, or you don't care at the current stage. So it is very important to have a understanding of what have been removed, or what is the task of the current algorithm. So it's not just always good to minimize the discrepancy whenever possible. Okay. So to overcome the limitations, we consider the following two strategies, or is the two principles that we want to follow. The first principle is that we want to have a way that to assess the alignability between the data set with certain statistical guarantee, where we can answer the question that is my data alignable after all. And secondly, we want to make the algorithm as transparent as possible, so that we have articulated inferential task. Basically, we know what have been removed after this applying this procedure, so that we can literally evaluate whether this is reasonable in a particular in a case-by-case basis. And for the rest of the talk, I'm just to introduce the framework that we proposed and some overview of the methodology, and then some applications follows. So the general framework of our consideration of alignment can be formally introduced by this noisy manifold model. It's very simple. So suppose we just have two data sets to be aligned or integrated together. The data set can be written as matrix yi, I equals to 1 or 2. And this is a n by p matrix, and the cell numbers. P is the feature numbers. And we suppose that this the high dimensional data set is a noisy observation of some intrinsically low dimensional signal matrix xi. And now in this case, what the batch effect means would be that there are some correspondence between the underlying signal matrices xi's. And the question is, what kind of transformations that is allowed to model the batch effect between two data sets? And now, considering this xi as some low dimensional object embedded in the high dimensional space, we can just start with some most simple transformations that we have good knowledge of in the Euclidean space, which consists of these 4 elementary transformations, including translation, just the mean shift, reflection, and dilation, or just the rescaling, and rotation. And we consider the transformations to be any combination of these 4 different kind of operations, which essentially makes this class of similarity transformations. And now, by focusing on this class of transformations that are allowed between the low dimensional structures, here we can define the alignable signals or perfectly alignable signal by the following: that for 2 underlying signals x1 and x2, there exists some transformation f in this similarity class, so that they are perfectly matched. And so this is the perfectly alignable, right? This is just a low dimensional structure that got rotate or reflect scaling after this similarity transformation, you got the other. Although what we observe is the noisy realizations of these 2 low dimensional objects, and also in the high dimensional space. And correspondingly, we can also define this partial alignability. Basically, it would be that there exists a subset of samples in x1 and x2, so that this relation holds. Okay, so now once we have this type of formulation of the batch effect problem, we can aim for 2 tasks. The first task is the test that we want to test whether 2 data sets are alignable. And under this framework, it essentially means that whether there exists some transformation f within this similarity class, so that the low dimensional structure is alignable. The other would be how can we find the best similarity map f in this class so that they are aligned. And here we have several challenges. First of all, this data set, as even in this model, we allow this high dimensional noise zi to be present. So we do not have exact observation of the signals. We have very noisy observation how to overcome that. Secondly, the data is high dimensional. We have thousands of genes to start with. We don't want this model to be imposed on some low dimensional space. This P should be of a much higher dimension so that this is more realistic. It turns out that this can be solved by a spectral method, which is a class of method that leverage the eigenvalue, eigenvector of some matrix carefully constructed from this problem. And it turns out that there is some mathematical theory called random matrix theory that characterizes the limiting behavior of the eigenvalue, eigenvectors, so that we can leverage that to control the uncertainty coming from the noise. So now something about the this test procedure. So again, since the null hypothesis here is that there exists some transformation so that this relation holds. And this transformation is one member from the similarity class, which contains an infinite number of components. This is essentially a composite null hypothesis. It's the problem itself is very difficult to solve, but once we recognize some geometric feature, we can much simplify this problem. So one thing that draws our attention is that there are some geometric quantity about this signal matrices that are invariant for any transformations within the similarity class. It turns out to be the eigenvalues of the signal matrix. So let's just recall what we know about the theory of principal component analysis, where in this case, the signal eigenvalues, the leading eigenvalues of the X matrix, it turns out in the PCA theory, it has interpretation to be the variation of the data across certain principal or second direction. Intuitively, you can consider that as the diameter of the data set along certain directions. And this eigenvalues will be just the theta 1 would be the principal direction, the variation of the data across the principal direction, and theta 2 would be the secondary principal direction. And in that way, we can easily see that this variation across certain direction that is invariant to a rotation to the data or translation or reflection. Okay. And in that case, it only needed to see that up to a global scaling alpha here, the two data set, the null hypothesis becomes that the two data set are the corresponding eigenvalues of two data sets are identical up to some global scaling that just comes from the scaling factor, nothing else. And our idea is essentially just to compare the eigenvalues of the two data sets to see whether they are similar or not. If they are similar, then they better have, they're more likely to have the same shape, although there could be some rotation, translation, and so on. Okay. Again, this is just a graphical illustration of the procedure. So suppose we have two scenarios. One is that the data are alignable up to some rotation, reflection, or translation. The other is it's not alignable up to that kind of transformation. And again, we obtain the eigenvalues of the two matrices and then compare whether they are similar or not. In that case, in the first case, they are actually similar. In the second case, they are very different. And from the random matrix theory, we can also derive a test statistic so that when they are similar, they are within some well-controlled non-distribution. When they are very different, then it will be detected. And then based on the p-values, we can just tell whether they are alignable or not. And again, this is for perfect alignment where all the eigenvalues are just all the leading eigenvalues are similar. But this can also be extended to so-called partial alignability, where we focus on subset of the data set to perform such a test. Okay, so here is just some very quick comments about the properties. We intentionally to keep the problem in the high dimensional space so that we do not assume the data have already been denoised or have already been projected to the lower dimensional space to perform all the subsequent tests. So that in that case, we directly operating on the high dimensional data set and we control the effect of high dimensionality so that we are not biased by dimension reduction method itself. Yeah, so we actually tested our proposed statistical test to a series of integration tasks. We considered 3 negative controls where basically we have clear knowledge about that these 3 pairs of data set are not alignable at all. And we just, here is just a demonstration of how the existing method align this completely unrelated data set. And you can see in many cases, they seems to be aligning the structures. And even just without the cell type annotation, it's very hard to discern whether the 2 data sets are alignable or not. Yeah, so for the 10 positive controls, we basically considered the benchmark data set that are commonly tested in the literature, which include that when data set are different in their sequencing technologies, like one based on SMART-seq, the other based on cell-seq, or about different samples, or about different tissues, or different experimental conditions, and so on and so forth. And in that case, these 2 data set, when they are coming from, say, similar type of studies, but with different sequencing procedures, in general, that similarity transformation group can account for the batch effect between the data set. So in general, this test prevents problematic integration, and it also has some suggestions that for many batch effect that have been, yeah, please go ahead.
Do you always test 2 data sets together?
Yeah, so for this paper, we mostly tested the pair of data sets for integration. But yeah, for test, it's currently just 2. But for actually in the next step, we're going to integrate 2 data sets. You can consider some natural ways that you can extend to, say, k different samples. You can either just do it sequentially, just the pairwise integrate them together, or actually we're considering some more general setup currently. But in this paper, it's mostly 2. I can come back to this at the end of the talk. Yeah, so a second message from this test is that it turns out that many benchmark across different kind of batch effect caused by different sources can be actually just accounted by the simple similarity transform. Nothing really complicated is needed to account for the batch effect. Indicate the goodness of fit of this simple transformation model of the single cell data. And now let's turn to this second step, is just the alignment of a pair of data set. Again, the goal is to search for similarity transform matching the low dimensional structures. And related to our previous formulation here, we're essentially solving this shuffled procrustes optimization so that for 2 data sets, X and Y, we are essentially looking for, so beta is the global scaling, R is the rotation of the feature space, gamma is the mean shift of that gene expression space. And here, because we do not have a previous existing correspondence between the cells across different data sets, so unlike the traditional procrustes analysis where we don't have this P, here we really have this sample permutation or sample linear transformation so that you can make connections between different cells across data set to account for this unknown correspondence. Main advantage of this alignment procedure is it is distortion free. So as you can imagine, we're just considering a similarity map for between data set. We do not locally transform we do not have local transformation that changes within data variation. So if you consider the low dimensional structure to be some shape like a triangle or square, it is perfectly preserved after you perform the transformation. And here we just take 3 benchmark data sets mentioned in the previous slides to compare different embedding or integration algorithms. The y coordinates characterize the preservation of within data structure before and after integration. Okay, and X coordinates is what people usually compare. It's just the batch effect removal. So our argument is that we not only just want to remove the batch effect to have a better mixing of two data sets, we also want to preserve the within data variations. In the extreme case, you can get perfect alignment. You just map everything to the origin, right? Everything get packed together. There is no batch effect at all, but you lose all the within data variations. Right. So in that case, we can see this procrustes analysis is pretty useful in preserving the within data structure, and it turns out to have some consequences in the downstream analysis. So here is just an example of the alignment based on the proposed algorithm for two data sets. And this is an example for where partial alignability is tested against, or a partial alignability is needed. As you can see here, there are part of the cell types that are not well represented by the other data set. And in that case, we can still preserve that difference between the two data sets while aligning the common parts. Here is just a example where we may suffer some distortion by using existing algorithm, which can much be alleviated using the proposed algorithm. It's just so for this particular gene, it is a marker gene for natural killer cell. Before integration, we can see that this is higher expressed in natural killer cell as contrast to other cell types. And after integration by this SMAI, we sort of preserve the differential pattern, whereas other algorithms just because of distortion of the expression level, in order to make the alignment, we lost that differential pattern. Here below is just a counter example where when the genes do not show any differential patterns, but other algorithms somehow create these artificial patterns after all. And this,
Just to clarify,
yeah, please.
the natural killer cell type is generated by each of the method, not pre-annotated.
It's pre-annotated. We take the cell types labels as the golden standard from this previous study, and just to see whether the differential pattern that we see before integration is still preserved afterwards.
And how that previous study, how did the gold standard was established? Like what methods do they use for that?
Yeah, so here we just, I think they use some annotation tools in general, and they have a list of marker genes. They based on that to identify cell types. Although, you know, I know there is a certain ingredient of circularity in the argument because we don't know in advance. But again, here we're not claiming that this should or should not be the marker gene. We just see that at least visually we see the differences before, and we hope that to be preserved afterwards.
I think my point of confusion is because you have the same cell type labels in all of these data.
Yeah.
Why would expression level change? You're looking at the same cell groups. Right?
You mean why would the expression level changes after the integration?
Right.
Yeah, so that's just for existing method. So including ours, the integration is about all the cells in the data set. And in order to integrate one data set to the other, there has to be some changes in the expression level.
So the normalization procedure basically shifts some things. Like
Yes.
the first picture that you
Yes.
showed us was a mirror, right?
Yeah.
You showed us that like there were.
Yeah, you can consider just a global rescaling. You divided everything by 2 to remove the batch effect. In that case, there is a differences. But in more complicated case, you may have some, you know, mean shift or gene specific changes. Yeah. Yeah, so yeah, for the time, for the interest of time. So basically, we also test the other downstream analysis, such as the spatial gene imputation. As you know, in order to impute the unmeasured genes in the spatial transcriptomic data, people usually bring together spatial transcriptomic data with whole genome RNA-seq data, and to use the common measured genes as an anchor to predict the unmeasured genes. And in order to do that, you again need to integrate the RNA, the measured gene profile across spatial transcriptomics and RNA-seq. And basically, we evaluate the different integration methods for that particular step of integration, along with the same subsequent analysis procedure. Essentially, we find that for that task, our algorithm have a pretty decent performance in prediction. And finally, one point we want to argue in the paper is that remember that in the alignment procedure, the progressive analysis, we literally solve for the mean shift, the rotation, and the scaling parameter for batch effect removal. And in that case, we basically know which gene has been changed or which particular what type of shift or rotation has been applied in order to make the alignment. And we can even examine this closely. For example, between these two data sets, one based on cell-seq, the other based on smart-seq. This is the mean shift vector that we solved from this optimization. It turns out that there are only a few genes whose expression measure being going through a global mean shift in order to align the two data sets together. And it turns out that some of the genes are just marker genes of some cell types within this data set. And as you can see here, can sort of give an explanation of what happens, what created the batch effect. For example, in this first column, this SST gene, we can see for this delta cell in the first data set is higher than all the other cell types. And in the second data set is also higher. But if you just look at the actual expression values, they are not comparable. So, but basically after a global mean shift, you do not change the global pattern, but it's just to match the corresponding values. It will make the data set more similar to each other. Yeah, some just a quick discussions of what we have been what we talked about. So basically, our procedure based on this explicit geometric modeling of the batch effect, it helps us to extend. Yeah, please.
We have a question from the chat.
Okay.
Oh, we can wait until the question section.
Okay. Yeah. So yeah, some related problem that we're also interested in and currently looking at is that, so first of all, the current procedure is linear. Just we look for a rigid similarity transformation. And in many biological processes, there are nonlinear components like differentiation or just lineage in general. And how can we extend the current framework to allow for nonlinear transformations? That's one thing. Secondly, since somehow this data integration is involved with low-dimensional embedding, right? We look for low-dimensional structure first, and in the meantime, we align that low-dimensional structure. So this identifying or learning the low-dimensional structure, again, is a key step in the workflow, and how to evaluate the uncertainty of any given low-dimensional embedding. Can we trust or not about low-dimensional embedding would be an issue of interest. And what we haven't talked about so far is how to integrate the current framework to multi-omic integration. It turns out that, so it happens that currently we're just considering rotation of object within the expression space. If you really have multi-omic integration, it will be useful to consider some latent shared low-dimensional space. So what we observed is some high-dimensional measurement of some latent low-dimensional object. But it's just the two kinds of views based on different realizations. And also another question would be, how can we incorporate the spatial information also within our picture? These are all very interesting questions that we want to explore in the next step. Yeah, basically this is what we want to share, and thank you for your patience and welcome to the questions. Oh, by the way, this is a co-author of the paper, and we have a GitHub page with some short tutorial about how to implement the algorithm.
Thank you so much, Rong. Yeah, so we can go ahead and just jump into the questions. I think folks are really interested in this topic. So we actually have one from Zoom. So please go ahead and just feel free to unmute.
Hi, Rong. I'm Nico from Malone Client's Lab. We spoke a lot about variation before. I love this kind of work. And I wanted to ask you about the criterion of looking into the spectral correspondence. You talk about, correspondence in the leading eigenvalues up to a certain scaling factor. And I wanted to ask you if you have an intuition, because a lot of the variation we see in single cell data is like the kind of variation you would see by sampling maybe the same individual many times. And then there's the variation that's included once depending on the kind of cohort you are sampling. So then kind of like you'll get some axis of variation become more representative if you include more diversity in a certain aspect of your cohort that you're studying. And I wonder do you have an intuition of how many eigenvalues like correspond to just the natural kind of like within individual variation, and how many would depend more on who you sample? Like this would be relevant in studies that maybe study the same tissue, but they one focused a lot more on kind of like inter-individual diversity, and therefore maybe the ordering of the eigenvalues might become a little more sensitive.
Yeah, you are absolutely correct. So one thing that so two things. First of all, by reducing the original, the testing for similarity transformation, the existence of similarity transformation to the test of singular values are already one step of reduction that we just try to improve the robustness of the algorithm. So if we just look at the similarity of eigenvalues, essentially we only want to measure whether the overall the diameter of the underlying shape from different directions are the same. So we allow certain structural discrepancy already, as long as it does not change the variation along those principal directions. That's first. Secondly, you're absolutely right that in some case, the order of eigenvalue might get changed. And that may comes from the variation of the experimental design that are intrinsic. In that case, unfortunately, if you consider like the full alignability of our current procedure, you're very likely to see that the algorithm tells you, no, they are not alignable at all. So to increase just the flexibility of the test, there are two things we can consider. One is that we can consider partial alignability, that we provide that option as a software implementation. Basically, the null hypothesis would be reduced, say that whether up to 50% of the cells that are alignable between the two data sets. In that way, it may provide some degree of flexibility so that you will not always reject the null hypothesis. And secondly, we are actually currently just looking at this problem, that how can we, in general, detect whether there are any sharing in the, say, the leading eigenvalue, or just in general leading eigen subspace up to some, change of orderings of the eigenvalues. It turns out to be a very challenging but interesting problem. So far, we do not have experimental result to show, but happy to discuss afterwards. And back to the first question, how to determine the number of eigenvalues. Currently, there are several very recent literature about this problem. It's just how to detect the significant eigenvalues from high-dimensional data. And in our paper, we point to one resources the algorithm called screen not, just the acronym for not using the screen plot that people always use. It's again that the algorithm is based on random matrix theory, which we find to be useful in many cases, but not, of course, not always useful. But it might provide some guidelines for us.
Thank you. That's a very thoughtful answer.
Thank you.
Any other
Yeah, maybe I ask two
Yeah,
questions
sure,
around
sure.
also the testing, which is very interesting, I think. I mean, like the two main problems what people typically ask with hypotheses tests, right, is on the one hand side, how reasonable is kind of like the null distribution assumption, right? And I think you say you derive the null distribution from random matrix theory. I'm not very familiar with that, but then the question is kind of like, do single cell matrices typically follow these random matrices, right? So this is my first question. And the second question is just this blue elephant in the room, right? P-value based decision making, you have a threshold. So when do you say we align yes or no? We align maybe at 0.04, but not at 0.06, but this is quite a discrimination.
Right.
So how do you, what are your recommendations there when in which kind of scenarios you say, oh yeah, here we go ahead and integrate as opposed to maybe not?
Okay, this is a very good question. Actually, I have a different answer to your two questions. So for the first question, I would say that random matrix theory currently, we find it has a great potential for analyzing single cell data. Some group in Europe and also at Yale, recently they published several papers basically pointing out this very surprising connection between random matrix theory, which is mathematical theory, to single cell experimental data. They find that for many kinds of single cell measurement, like gene expression or even like chromatin accessibility, many kinds of sequencing data, just after a very simple but specific kind of transformation or normalization of the counts, they will see just making a histogram of, let's see, eigenvalue distribution of the data matrix. It aligns perfectly with the theoretically derived limiting laws based on, say, Gaussian distribution. It turns out, so the conjecture currently is that there are some universality of this random matrix theory, although it's just derived based on Gaussian, but it's just a very, there is a great conformity between the experimental data and the theory. We also, this also is kind of motivation for us just to use this random matrix theory. And secondly, there are mathematical breakthroughs recently just proving the universality of many random matrix theoretical results. It shows that many limiting distributions derived in random matrix theory are independent from the specific distribution you start with for the individual entries. It's like central limit theorem. It's just no matter what you start with, as long as certain normalizations are done, then what you observe in the data, it can be universal. So we find that to be a very powerful ingredient for the analysis. But that, again, back to your second question, that does not make my argument that saying that now I still think the hypothesis testing framework is always good. So if I do it again now, I would, instead of considering proposing a test with a p-value for telling people whether your data is alignable or not, I would instead propose a measure or metric or score like between 0 and 1, so that to tell you that whether, to what extent your data, it's very easy to get that. Basically, you can consider a distance measure based on the similarity of eigenvalues. Again, using some theory, you can come up with a very decent score, so that you have a quantitative continuous measure of the alignability. So hopefully that will be implemented in the future work. And back to the previous question about extension of the current framework to multiple data sets. For test, it's a little bit hard for now. But for alignability, since we use procrustes analysis, there is a natural generalization called generalized procrustes analysis existing in the literature. So that is one possibility to extend the current framework. But the downside is that current procrustes analysis for two data sets, it admits closed form solution. So you don't have to optimize very computationally intensively. But for general K data set, the generalized procrustes analysis does not have that clear mathematical formula you can leverage on. So that's the pros and cons.
And maybe an implementation question. around
Yes,
it.
yes.
So we tried it out. Actually, Daniela tried it out. Works super well. The implementation on GitHub. It's an R package, right? It's obviously very useful. Just wondering, do you have any plans to kind of like maybe put this CRAN?
Yeah. That would be nice. And even at least like for algorithmic steps that needed for multi-data integration. I have some ideas of how to do so, but just haven't had time to do so. But very happy to collaborate on any.
All right, I think we're at time. So thank you again, Rong, and we'll give Rong one more round of applause here. But also, if anyone is interested, Dr. Ma has graciously agreed to just take a little bit of time to talk more in depth about any of these topics. So anyone on Zoom, feel free to stay on as well.
Yeah.
We can discuss more. Thank you.
Thank you.
I had a question actually. So there's a lot of kind of like methods now that kind of do like imputation for any like missing like features or non-overlapping features. So I was wondering like what your suggestions might be if you, for instance, like do the SMAI test and it's like not alignable. Would you suggest something like first doing imputation first and then retrying, or like what are your thoughts about that?
Right. So current paper, we tested this first test for alignability. If it looks so, then we can use the, I mean the test for just the overlapping part, and then try to do the imputation. But again, I think in general, it should not be the case that we make our decision purely based on a p-value. So if possible, I would like to report again a score so that it does not literally affect what you want to do eventually, but you just have an outside, just additional information. Even say your alignability still is low, but you still can apply, say, the SMAI align. There is nothing harmful for you. It's nothing prevent you from searching for the best similarity transformation between the two data sets. And sometimes the low alignability may not be something that is uninteresting. It may be, say, across disease and healthy samples, some differences may be exactly what we want to see after aligning the, say, the first order differences, like mean shift or whatever. So this low alignability could be just a flag over there that you can take into account, but you still can do the downstream analysis.
Yeah, I imagine it's very biologically question driven.
Yeah. So I guess this is just some computational support that you can call for, but nothing serious.
So earlier, in the talk, you mentioned that you were trying to integrate CNV profiles with gene expression.
Yeah.
I think it's very interesting. I work on CNV a lot.
Okay.
And I wonder what's the goal of aligning those profiles, because
Yeah.
for CNVs, you can only have like up to 5 CNV profiles
Right.
per data set. I don't think it's a huge feature set to work with.
Yeah, it's not diverse for each individual entry. Yeah, so in that case, the goal is a little bit different. So in that context, the main problem interest would be the cancer cells, how to characterize that. And basically, if it's normal cells, then there's nothing variation in CNV. So their experiment, they have single cells captured from tumor, which basically contains tumor cells and stromal cells, or just normal cells within the tumor. And in that case, you can imagine if you look at the CNV profiles, you can have, say, 3 big clusters containing 2 kinds of sub-tumor clones and 1 normal cells within them. And on the other hand, you will have more clusters based on the RNA expression because there could be tumor clusters, but for normal cells, it could be different cell types. So they will have more clusters. So the biological question of interest for them would be, first of all, whether if you just focus on the tumor cells, which you can determine just based on the CNV, right? And the cell IDs, you know what cells are tumor cells. You want to see whether the cluster patterns based on CNV matches the cluster patterns in expression. This is what people expected, right? You have different CNV, then you have different RNA expression. That is also the basis for many algorithms that infer the CNV based on the gene expression. But in their analysis, they actually find, so of course, this is true in general, but they also identified some small cluster, identifiable small clusters where, say, they show clear differences in the CNV profile. I think they find something happening in the X chromosome, and clear difference over there, but very similar expression profiles at the end. And there are some, I've seen some other studies even find some clusters that they are allegedly just for sure cancer cells, but their expression profiles look very similar to a kind of normal cell. Although this is a relatively small clusters, does not change the overall picture, but to them, they really think that outliers or just something does not follow our expectation to be biologically very interesting. So in Mike Wheeler's group, they call it the spy cells, so that they have very abnormal copy number variations, but expression looks very similar to a kind of stromal cell. So you cannot find that without having this multi-omic data, right?
You only need multi-omic data to do that.
You really need to have this label correspondence in order to identify.
I agree with that, but I don't know how alignability, like virtual, comes into play in the analysis of this kind of.
Yeah, so our yeah, you're right. So our current formulation, the current algorithm does not allow us even to consider different type of features, right? We have to have this list of genes. But for aligning CNV, you have beans, right? The gene you have RNA expression of genes, the different kind of features. Then really need to develop some alternative or extensions. Currently, we have some idea, just a rough idea of how to do so, but yeah, it needs some time just to evolve. Yeah.